this, but this is very much relevant to our discussion here. So I want to show you here the salicylate interaction with a lipid bilayer. Okay, and uh, and here you can see this movie, which is like the aspirin derivative, how it is interacting with the lipid bilayer membrane. And you can see that they're moving around in this across the uh, on the outer plasma membrane, if you consider this, and this is the inner plasma membrane. And you can see that with time, they basically, there is an equilibrium where the adsorbed molecules on the outer membrane and inner membrane basically become equal. Okay, you can see right here. So now the question is, can we probe such processes using surface sensitive SHG? If you recall that I showed you that SHG is a surface sensitive technique, and if you have a ordering, okay, you can, that's a ideal system to study the surfaces. And can we monitor this kind of processes using complex surface interfaces using SHG technique? Okay, and sure we can. And here I want to show you our recent results on looking at time dependent dynamics in lung cancer cells, single cells. Okay, so here there's a lot of information, and I want to explain you from here. You can see um, so what we did is we did a SSG microscopy where we add this SSG dye, okay, which gives you a is a good SSG scatterer, this malachite green dye. Okay, it's actually here it's a derivative, isothiocyanate derivative, and it has been known that it has this triphenyl methane type of backbone structure, and it is also known to be a marine antibiotic. So it has implications to look at this drug, how the drug is basically transporting across the cell membrane. So this is a good system to study, and you can see here at 0 minutes when we add these dye from the side, okay, you don't have any cell visible, okay. Um, but as this dye basically gets adsorbed onto the surface, okay, so this is a cationic dye, and the phospholipid membrane is negatively charged, so you have this electrostatic interaction getting adsorbed, and they get aligned, okay, and that leads to the increase in the SHG signal as the dye is adsorbing onto the surface, as you can see in this cartoon here. Okay, and at around so you can see that as in the images that after 25 minutes you see this this cell is visible now. Like you can see these bright signatures coming from SHG due to the adsorption of this dye, malachite green dye, onto the plasma membrane or outer leaflet of the plasma membrane. Okay, and at 34 minutes we see the maximum intensity, and after that we see this drop in intensity in the SHG channel. Okay, and that's due to that you have a monolayer adsorption, or you have a maximum adsorption, and you can which is directly proportional to the number of molecules that is outside, okay, or outer side of the plasma membrane. And what happens is that, like, when as the dye molecules translocates through this lipid bilayer membrane, so this distance is just like 5 nanometer, okay, a very small distance, and then when they go on the other side, they get aligned again opposite to the orientation on the outer surface. Okay, so there is a net cancellation of dipoles, or you can say that it is destructively interfere, and as I mentioned, that leads to the signal drop in SSG. Okay, due to this they get exactly oriented in opposite direction, as you can see in this cartoon illustration, where positive charge represents this malachite green dye. And this is basically a different region of interest. You can we did also the averaging, and you can see that here. You can also get the transport rates and the adsorption time. We saw that around 34 minutes for malachite green dye, and the transport lifetime, the rate which gives you the transport lifetime across the plasma membrane is around 8 minutes. So you can monitor the rates of these molecules translocating across the plasma membrane, okay, using SHG technique. And also precisely the number of molecules can also be found. And here I'm showing you further, like we also looked at the functional group dependence, this MgITC versus Mg. So due to the incorporation of this isothiocyanate group, you have a much larger dipole moment in this, and that gives you the much stronger affinity to binding onto the surface of the plasma membrane. And you can see that from this image here, we basically see again similar behavior, and the adsorption time was much lower for MgITC versus Mg, where Mg requires around 75 minutes, nearly more than double, okay, with respect to MgITC. And also, we not only did live cells, but also we looked at the fixed cells. So what we found using this MgITC, where we use the same dyes, and we saw that in a fixed cells or dead cells, the adsorption and transport is much longer. Okay, so you can get the rate constants from this, and we also see these subtle fluctuations in the intensity, which we are currently I'm working on the analysis, which we think is it due to the experimental artifacts associated with the instability in the laser, or it is due to some complex uh, biological function of the cellular metabolism. So this work is still in progress, and now we are not, of course, the first one to study this, and this idea was introduced first by Ken Eisenthal's group, who is one of the pioneer in the surface sensitive spectroscopic studies. And in late 1990s, using the synthetic liposomes, he basically showed that due to the adsorption of these malachite green dyes, you get a second harmonic adsorption, second harmonic scattering signal due to this adsorption from this malachite green dye, because again, due to this coherent summation of this electric fields from individual oscillating dipoles, right? They get adsorbed, you have ordering, and you see a enhanced SHG response. And later on, this was studied in a gram-negative bacteria, which has antibiotic resistance because it consists of two compartments. Okay, there is an outer compartment as well as an inner compartment. So the molecule has to not only pass through the first outer plasma membrane, but also this periplasm region in between, and also has to travel much longer distance and get across the inner plasma membrane. And they can see this behavior in their SSG intensity time traces. You can see that the first, when you add this malachite green dye, you have a very sharp rise in the SSG signal. Then as the molecule gets aligned oppositely on the inner leaflet of the plasma membrane, you have a sharp drop in the signal. And then again, due to the adsorption behavior, you have a second rise, and then you have some constant intensity, okay, in the SSG channel. And later on, very recently, this work was published, and here they adapted this experiments in, again, gram-negative bacteria consisting of two inner as well as outer plasma membrane, and they with added pH effects, okay, in different pHs, they showed that they studied this, again, seeing a similar behavior, okay, and the two-step process, like adsorption, transport through first plasma membrane again, and the second one. And also this group here from Calhoun's group recently also studied in a gram-positive bacteria, which only has one membrane compartment. And as you can see that when you have this incoherent, like randomly oriented molecules, you don't have any SSG, but when they get adsorbed onto the plasma membrane, you have a SSG signal rising, and then as they translocates across the plasma membrane, get aligned in the opposite direction, the net cancellation of dipole moments lead to the decrease in the SSG signal. They also looked at the two-photon fluorescence also, but I won't go into the details. But most of these studies were done in bulk spectroscopic measurement. What I mean by bulk is basically they were done in the cuvette. Okay, but what our approach was like looking at the single cells where you have uh, when you do single cell experiments, you have heterogeneity also, and you need to also study that in much better detail, and also